Good morning class, welcome back to microbiology. This is Silvana O'Coney and today's lecture, lecture 2, is about prokaryotic cell structure and function. One of the most important prokaryotic structures is the bacterial cell wall. The cell wall is a rigid or semi-rigid structure that lies just outside the plasma membrane. Those prokaryotes that lack cell walls have other features that fulfill the cell wall function. Functions of the cell wall. The cell wall provides characteristic shape to the cell, protects the cell from osmotic lysis, and in some groups of bacteria, the cell wall may also contribute to pathogenicity. In 1884, a Danish bacteriologist called Hans Christian Gram developed a technique that divided bacteria into two major groups based on the response of those bacterial cells to a procedure called the Gram stain procedure or Gram stain reaction. Bacteria that stain purple as a result of this reaction are called gram-positive bacteria, and those that stain pink are called gram-negative bacteria. Now, Hans Christian Gram devised his technique not for the purpose of distinguishing one type of bacterium from another, but to make bacteria more visible in stained sections of lung tissue. He published this method in 1884 and included in his short report the observation that the typhus bacillus that he was studying did not retain the stain. The staining reaction is due to differences in the cell wall structure between these two groups of bacteria. But these structural differences did not actually become clear until the advent of the transmission electron microscope many years later. Let's now look at the structure of the gram-positive cell wall to the left of screen and the gram-negative cell wall to the right. The gram-positive cell wall consists of a single 20 to 80 nanometer thick homogeneous layer of a substance called peptidoglycan, and this wall lies just outside the plasma membrane. In contrast, the gram-negative cell wall is quite complex. It has a 2 to 7 nanometer peptidoglycan layer covered by a 7 to 8 nanometer thick outer membrane. So because of the thicker peptidoglycan layer in gram-positive cells, these gram-positive cell walls are more resistant to osmotic pressure than those of gram-negative cell walls. The space in between the plasma membrane and the peptidoglycan layer in gram-positive cell walls is called the periplasmic space. If you look at the gram-negative cell wall, you'll see two periplasmic spaces, one between the red plasma membrane and the peptidoglycan layer, and another space between the peptidoglycan layer and the outer membrane. The nature of this periplasmic space in gram-positive and gram-negative cell walls is quite different. Just note also in the gram-negative cell wall that the peptidoglycan layer and the outer membrane are together referred to as the cell wall. Now let's take a closer look at the structure of peptidoglycan, also known as murine. Peptidoglycan is a polymer, it's a mesh-like polymer which contains many identical subunits. Each of these subunits contains two sugar derivatives and several different amino acids. The first of the two sugar derivatives is called N-acetylglucosamine. The other is called N-acetylmuramic acid. We can abbreviate those two sugar groups to NAG, NAG, and NAM, NAM. The chains of linked peptidoglycan subunits are cross-linked to each other by peptides, that is, short stretches of amino acids. Here we have an illustration of a single subunit of peptidoglycan. This particular peptidoglycan monomer is found in E. coli bacteria and also in most other gram-negative bacteria. So we can see at the top of screen one NAM and one NAG. And these are the two sugar derivatives that look very, very similar to glucose. The sugar group to the left, NAM, is then attached to a chain of 4 amino acids, a peptide chain of 4 amino acids, and they're actually connected to the carboxyl group of the NAM residue. Now, what's interesting is that 3 of these amino acids are not normally found in proteins. That is D-glutamic acid, D-alanine, and diaminopimelic acid. You can see, due to the prefix, that these are D-isomeric forms. Normally, amino acids are L isomers. So the presence of these D amino acids protects the peptidoglycan from degradation by most peptidases. Peptidases are enzymes that generally cleave proteins or peptides. And these peptidases generally only recognize L isomers of amino acids. So this makes peptidoglycan quite a tough substance. In order to make a strong mesh-like polymer, chains of linked subunits have to be joined by what are called cross-links between the amino acids. And this can be done in one of 2 ways. So at the top we can see an example of 2 E. coli peptidoglycan subunits, and they're directly cross-linked between the alanine of one subunit and the DAP amino acid of the second subunit of peptidoglycan. So this is what would normally occur in many of the gram-negative bacteria, including E. coli. In B, we can see a different way in which the peptidoglycan units are cross-linked. And this cross-link is mediated by what's called a peptide interbridge. That is just a short peptide which consists of 5 glycine amino acids linked together, and this interbridge, if you like, connects the alanine from one peptidoglycan subunit, the one on the left, to the lysine amino acid of the second peptidoglycan subunit. And this is what is observed in Staphylococcus aureus peptidoglycan. Here you can see the resulting mesh-like polymer. This mesh is dense, strong, porous, but a very flexible network. Now let's take a closer look at gram-positive cell walls. Gram-positive bacteria have cell walls that are thick and composed primarily of peptidoglycan. They also contain large amounts of an acid called teichoic acids. Some gram-positive bacteria have a layer of proteins on the surface of the peptidoglycan. So for example, Staphylococci have these attached proteins that are involved in interactions of the cell with the environment. And some of these proteins even have roles in virulence, such as aiding in adhesion of the bacterium to host cells. Pictured here, we can see an isolated cell wall from the soil bacteria Bacillus megaterium. And just to get some idea of size, just note these latex spheres, which are 0.25 microns in diameter. The periplasmic space of gram-positive bacteria lies between the plasma membrane and the cell wall and is generally much smaller than that found within gram-negative bacteria. Gram-positive bacteria often appear to lack a periplasmic space, and the fluid within this space, called the periplasm, seems to have relatively few proteins within it, probably because the peptidoglycan is quite porous, so any proteins secreted by the cell usually just pass straight through it. Enzymes secreted by gram-positive bacteria are called exoenzymes, and they often function to break down nutrients in the environment that would otherwise be too large for transport across the plasma membrane. Here we can see the relatively simple structure of the gram-positive cell wall. Note in purple the teichoic acids, which are found embedded in the peptidoglycan layer of the cell wall. And also note the lipoteichoic acids, which attach the cell wall to the plasma membrane. Now let's look at gram-negative cell walls. These cell walls consist of a thin layer of peptidoglycan surrounded by an outer membrane. This outer membrane is composed of lipids, lipoproteins, and lipopolysaccharides, abbreviated to LPSs. We'll come back to these. Gram-negative cell walls lack teichoic acids. So gram-negative cell walls tend to be more complex than those found in gram-positive cells. The peptidoglycan layer is very thin and may constitute only about 2 to 5% of the cell wall weight. In E. coli, in fact, it only contains 1 or 2 sheets of peptidoglycan. The periplasmic space also differs from that found in gram-positive cells. The periplasmic space may constitute as much as 20 to 40% of the total cell volume. And unlike the gram-positive cell walls, there are many enzymes present in the periplasm of gram-negative cell walls. The outer membrane of gram-negative bacterial cells lies just outside the thin peptidoglycan layer. It's actually linked to the cell in 2 different ways. The first is by a protein called Braun's lipoprotein. This is the most abundant protein in that outer membrane. And this lipoprotein is covalently linked to the underlying peptidoglycan, and it's embedded also in the outer membrane by its hydrophobic end. The outer membrane and the peptidoglycan are so firmly linked together by this lipoprotein that they can actually be isolated as a single unit. The second linking mechanism involves many adhesion sites which directly join the outer membrane and the plasma membrane itself. These 2 membranes appear in some areas to be in direct contact with each other. Here we have a nice illustration of the gram-negative cell wall. Let's start at the bottom of the screen, and you can follow the labels to the right-hand side. So we start with the lipid bilayer of the plasma membrane, and we can see the phospholipids, of course, and the integral membrane proteins. Okay, just above that we have the first of the periplasmic spaces, followed by a very thin layer of peptidoglycan, and then the second of the periplasmic spaces. And in this second periplasmic space we can see the linking of the peptidoglycan with the outer membrane by those blue bronze lipoproteins. Okay, just to the outside of that we can see the outer membrane, also a lipid bilayer, but we can see that there are different proteins inserted into the outer membrane. These are called porins, and we can also see what looks like fine hairs on the outside of the outer membrane, but these are in fact lipopolysaccharides, which we'll come back to in just a moment. The most unusual component of the outer membrane is the lipopolysaccharide, or LPS. These are large complex molecules that contain both lipid and carbohydrates and consist of three parts. The lipid A component, which is shown here in green, the core polysaccharide, shown in blue, and the O side chain, otherwise known as the O antigen, shown in purple. This is the Salmonella lipopolysaccharide, which is the best studied of the LPSs. The fatty acid component of the lipid A, shown again in green, attaches to the outer membrane, while the remainder of the LPS projects from the surface of the outer membrane. The core polysaccharide, in light blue, is constructed of 10 different sugars, most of them quite unusual in structure. LPSs have several important functions. Let's look at the first one. LPSs appear to play a role in protecting the cell, the bacterial cell, from the host defences. The O side chain of the LPS, also called the O antigen, because it elicits an immune response. This response involves the production of antibodies that bind to the strain-specific form of LPS that elicited the response in the first place. However, many bacteria can rapidly change the antigenic nature of the O side chain, thus avoiding the animal host defences. Importantly, the lipid A portion of the LPS is often toxic. As a result, the LPS can act as an endotoxin and cause some of the symptoms that arise in gram-negative bacterial infections. So if this type of bacterium enters the bloodstream, the LPS endotoxin can cause a form of septic shock, which is very, very difficult to treat. Another important function of the LPS is that it contributes to the negative charge on the cell surface. This is mediated by the core polysaccharide, which contains a negatively charged sugars and phosphates. A major function of the LPS is also to create a permeability barrier that prevents the entry of bile salts, antibiotics, and other toxic substances that might kill or injure the bacterium. Despite this role of LPSs in creating a permeability barrier, the outer membrane of gram-negative bacteria is actually more permeable than the plasma membrane, and it permits the passage of small molecules like glucose and other monosaccharides. This is due to the presence of porin proteins, which are found in the outer membrane. Larger molecules such as vitamin B12, also can cross the outer membrane, but these large molecules don't pass through porins. Instead, they're transported across the outer membrane via specific carrier proteins. Now that we understand the differences in cell wall structure between gram-negative and gram-positive bacteria, we can start to understand the mechanism of gram staining, which distinguishes between those two groups. It seems likely that the difference between gram-positive and gram-negative bacteria is due to the physical nature of their different cell walls. It's important to note that the peptidoglycan itself is not stained. Instead, it seems to act as a permeability barrier, preventing the loss of crystal violet stain. So during the gram stain procedure, the bacteria are first stained with crystal violet and then next treated with iodine, which promotes retention of the crystal violet dye. When gram-positive bacteria are then treated with ethanol, the alcohol is thought to shrink the pores of the thick peptidoglycan. Therefore, the crystal violet dye iodine complex is retained during this step and the bacteria remain purple. In contrast, remember gram-negative peptidoglycan is very thin, it's not as highly cross-linked, and it has larger pores. So the alcohol treatment may extract enough lipid from the gram-negative outer membrane to further increase its porosity. So for these reasons, the alcohol more readily removes the purple crystal violet iodine complex from gram-negative bacteria in comparison to gram-positive bacteria. After that, the gram-negative bacteria are then easily stained red or pink using a counterstain called safranin. The gram stain was developed in 1884 by the Danish physician Christian Gram, and it's the most widely employed staining method used in bacteriology. The Gram stain reaction is an example of differential staining. These are procedures that are used to distinguish organisms based on their staining properties. Use of this Gram stain reaction divides bacteria into two classes, gram-negative and gram-positive. Let's look at the steps involved in gram staining. In step 1, the bacterial smear is stained with the basic dye called crystal violet. This is the primary stain. This is followed by treatment with an iodine solution in step 2, and this iodine solution functions as a mordant. The iodine increases the interaction between the cell and the dye so that the cell is stained more strongly. The bacterial preparation is next decolorized in step 3 by washing with ethanol or acetone. And this step generates the differential aspect of the gram stain. That is, gram-positive bacteria will retain the crystal violet, whereas gram-negative bacteria lose their crystal violet and become colourless. Finally, in step 4, the bacterial preparation is counterstained with a simple basic dye, different in colour from crystal violet. Safranin is the most commonly used counterstain, and it colours gram-negative bacteria pink to red, but leaves gram-positive bacteria dark purple. So the end result is that gram-positive cells remain purple, gram-negative bacterial cells appear pink or red.